Good evening, everyone. It's my pleasure to welcome you to this evening's ASI Let's Grow webinar titled Integrated Parasite Management Strategies for Sheep Producers. My name is Jay Parsons. I'll be your host for this evening. I am a professor in the Department of Agricultural Economics and the operational leader for research in the Center for Agricultural Profitability at the University of Nebraska-Lincoln. This webinar is brought to you by the American Sheep Industry Association, and we want to start off by just giving them a shout out of thanks for their support and encourage you to visit the ASI website to learn more about the sheep industry and to access the large volume of resources that they have available there to help you be successful in the sheep business. The ASI website URL is www.sheepusa.org. If you're not currently a member of the American Sheep Industry Association, I'd like to encourage you to become one. Most people do that by joining their state sheep association. A list of the contact people for those associations is available on the ASI website under the contacts link. There's also a join link at the top of the page where you can read more about association activities and the benefits of ASI membership. I'd like to remind all of our listeners that this webinar is being recorded. All webinar registrants will receive a follow-up email within the next 48 hours with a link to the webinar recording, as well as a link to access the webinar slides. The links will also be posted on the ASI website in the Let's Grow educational materials. We are slated for about a 45 to 50-minute presentation this evening, followed by about 20 minutes of question and answers with our presenter. Feel free to submit questions at any time, including during the presentation, by typing them into the question dialog box at the bottom of your control panel. I will monitor these questions throughout the evening and moderate them to our presenter during the Q&A session after the presentation. You will also have the opportunity during the Q&A session, if you would like, to unmute yourself and ask your question directly to the speaker. I'll go over those instructions when we start the Q&A session after the presentation. So it is now my pleasure to introduce this evening's speaker, Dr. Andrew Weaver. Dr. Weaver is an assistant professor and extension small ruminant specialist at North Carolina State University. His research and extension interests focus on utilization of genetic tools and management practices to improve parasite resistance, performance, and the end product value of small ruminants. Prior to joining North Carolina State University, Dr. Weaver studied under some of our best land-grant university research, teaching, and extension specialists in the small ruminant area. He earned his bachelor's degree at Michigan State University, where he also participated in the meat judging team and worked at the sheep teaching and research farm. After that, he completed a master's degree at Virginia Tech University, where his thesis topic was a study of terminal sire options for hair sheep producers. And finally, he completed his PhD at West Virginia University, where he gained valuable experience working in Dr. Scott Bowdridge's parasite immunology lab and completed dissertation research on the impact of sire selection and breed on parasite resistance in sheep. So it's with great pleasure that I turn the floor over to Dr. Weaver for this evening's presentation. Welcome, Dr. Weaver.
Thank you. Thank you for the introduction, and great to be here this evening. Thanks for having me. So this evening, I'd like to talk about parasite management, and I know this is kind of a hot topic and one that as an extension specialist, I get asked about on a fairly frequent basis. So I wanted to go over some of the management strategies that we have for parasites. But first, I wanted to touch on parasite biology. And I think first we have to understand what the parasite is that we're worried about, what that parasite does, its biology, so that we can understand how to manage that worm and tools that we have in our parasite management toolbox to help combat that infection and the parasite burden that our animals could be exposed to. And so there's a lot of worms that could infect our animals. Screen working here? There we go. Lots of different worms that can infect our animals. A lot of them fall in a family of parasites called strongula nematodes, and that includes Homunculus contortus, Telurdossagia, Ostratasia. If you're familiar with cattle parasites, that's a fairly common one. Other Trichostrongylus species, Capiria nematoderius. A couple of these, such as Homunculus, are more popular or more common, I should say, in areas of the country like the Southeast US and in hot, humid climates. Some of these other parasites more commonly seen overseas and a a bigger challenge to the sheep industry in other parts of the world. We also have to worry about protozoan parasites like Eimeria. So we think about the parasites that cause the most challenge or the most prevalent, cause the most issues in our sheep. We really focus on Homunculus contortus and Eimeria or Coccidia. And for today's presentation, we'll focus primarily on Homunculus and these Strongula nematodes. I did wanted to share this chart here, which is some survey data from the upper Midwest of parasite populations on a number of sheep farms. And you can see here just the prevalence of Homunculus in those parasite populations. Even in an area of the country like the upper Midwest, where it's maybe a little bit more temperate than what we experience here in the Southeast, Homunculus is still the most prevalent parasite in their population. And so this worm is the one that we are most concerned about. By and large, it's going to cause most of our problems, and is the one that we place a lot of focus on. And so that's why for the remainder of today's talk, I'm going to focus primarily on Homunculus or the barber pole worm, although the management strategies that we discuss and the tools that can be utilized are relevant for a number of our other parasites as well. So Homunculus contortus, this worm is a blood feeder. It actually uses the little tooth-like structure that you can see right here. It's called the lancet. This tooth-like structure, it slashes at the walls of the abomasum, that fourth compartment of the ruminant stomach, causing blood release. And when that blood is released, it then consumes that blood, and that's what it feeds on. And it gets its name, the barber pole worm, from the fact that when it consumes that blood, the blood-filled intestines wrap around the reproductive structures of the adult female worm, resulting in this barber pole appearance that you see in the image on the lower right. And so that's where it gets the barber pole name from. As a result of this blood feeding, it causes anemia in the host. As a result of the anemia, we see decreased performance in terms of weight gain, wool production, milk yield, and traits like that. And then we see death in severely infected individuals. This is a highly prolific parasite. A single female can release thousands of eggs in a given day. And one fun fact about this worm, that single female can actually mate with multiple males. And so not only is she releasing thousands of eggs in a given day, but those eggs could be fertilized by multiple males. And so when we think about the opportunities for genetic diversity, and we think about things like dewormer resistance and the occurrence of that, there's a lot of opportunity. And so we need to be aware and cognizant of that. Due to the impacts that this worm has on our sheep populations, it does result in millions of dollars in economic losses annually. So anything that we can do to help manage this parasite, better manage this parasite, and minimize the infection level in our animals is going to improve their performance, their fitness and survivability, and improve our bottom line as well. When we think about managing the parasite, we have to first understand the life cycle. The life cycle is very important to this, and by understanding the life cycle, we can apply a lot of different management strategies to help mitigate the opportunities for animals to get infected. And so I wanted to walk through the life cycle here. And so we'll start off at the egg stage. So those eggs are deposited in the feces of the sheep. Those eggs, within a day or two, hatch into what we call L1 larvae, that first stage larvae. And those larvae reside in the environment. They consume bacteria and grow, and as they get bigger, they eventually molt and enter what we call the L2 stage. The L2 stage pretty much does the same thing as the L1 stage. They eat, they hang out, they grow, and eventually they molt again into the L3 stage. Now, the L3 stage is very important. The L3 stage is the only infective stage to the host. So the sheep can consume as many L1, as many L2 larvae as they want, and they'll never get infected. It's only the L3 that can infect the host. And the development of this parasite, or the life cycle of the parasite, does not continue past the L3 stage until it's inside the host. So they get to the L3 stage and they hang out and they wait to be consumed before they continue on with their life cycle. Another unique characteristic about the L3 is it actually has a secondary covering over it, a secondary sheath that protects it from harsh environmental conditions, but it also prevents the worm from being able to feed. And so they're storing, during the L1, the L2 stage, they're storing up energy reserves. When they get to the L3 stage, whatever they have when they get to the L3 stage is what they've got. They're not going to get any more because they're not feeding. And so they're stuck with that. So anything we can do to extend the lifespan or extend the time those L3s are out on our pastures before they're consumed is going to increase the chances of the L3s using up those energy reserves, not having energy left, not being able to feed, and eventually dying. And so we'll come back to that in just a little bit as one of our management strategies. So the L3 is hanging out in the environment. It's got that limited lifespan, but it's going to maximize its chances of getting inside of the host. So it's going to hang out in the dew droplets on the grass, and it actually rides those dew droplets up and down the grass each and every day. And so when we think about when the dew is out on the grass, it's generally the later evening hours, overnight, the early morning hours, and then during the day when the sun gets higher in the sky, that dew is going to reside. Well, when we think about when our ruminant livestock are grazing, they're grazing during the cooler times of the day, the later evening, overnight, and the early morning hours when that dew is up on the grass, and the chance of exposure to that parasite is the greatest. And so really the parasite's figured out a way to maximize its chances of being consumed, continuing on its life cycle. And so it's going to hang out in those dew droplets. It's going to be eventually consumed by the sheep. Once it's consumed, it makes its way to the abomasum. It molts into the L4 stage. Fun fact about the L4 stage, the L4 larvae, they do feed. They feed on blood just like the adults, but they are not sexually mature and they do not produce eggs. And so we could have L4s inside of our animals. We could have L4s consuming blood, causing anemia, causing all of those symptoms that we talk about. But if we were to take a fecal egg count, we wouldn't find any eggs in that sample because the L4s are not mature yet. And so the L4s are going to reside there in the abomasum, continue development, continue to grow, and eventually mature into the adult worm that is sexually mature and does start producing those eggs. From the time the L3 are consumed by the host until we get those eggs in the fecal sample is about 21 days, about 3 weeks. So there's essentially a 3-week lag time between when the parasite's consumed from the environment to when we can actually observe the parasite eggs in our fecal sample. So we need to remember that. We need to remember that, what we're seeing in a fecal today is really maybe what they consumed 3 weeks ago, or even more than that. It's not what they consumed yesterday. So keep that in mind. So about 21 days inside the host from L3 to eggs, and outside the host, that time frame is highly variable. I say generally 7 to 14 days during really hot, humid, wet weather. It could be even shorter than that. It could be 3 or 4 days. During cooler times of the year, maybe it's drier. It could be longer than that. And so that time frame is relatively variable, but generally in that time span of a week or two or so. Another thing to keep in mind is that the adult larvae, or the adult worm, does not bind the epithelial surface of the abomasum. If I can get this video to play here, there we go. So you can see those adult worms with that barber pole appearance and the adult females. You can see those worms moving around the epithelial lining of this abomasum. And those worms are not attached to that lining. They have to swim, they have to continue motion to maintain their position within the gut. So this is very important when we think about how our dewormers work, how the sheep's immune system works to help mitigate that parasite infection. Anything that prevents those parasites from moving is going to result in their expulsion from the GI tract and their removal from the host, removal from that sheep. And so we'll come back to this as well, but it's a common misconception that these parasites attach. They do not attach. They have to maintain that swimming motion to maintain their position within that gut. Another interesting characteristic about the worm is this hypobiosis, this arrested development, the stage that they enter when the parasite senses that the environmental conditions are not favorable for continued development. So as we enter the winter months, when the chances of larval survival in the environment is quite low, or times of extreme drought, when the likelihood of that life cycle being able to be completed is low, this parasite is able to enter this stage of hypobiosis, this arrested development, where they hang out at this L3, L4 stage inside the host. They pretty much set up camp and hang out until the environment becomes more favorable for their continued development. They resume that development when some signals suggest that the environmental conditions are more favorable. Now, this process is not well understood. There's a lot of work still to be done in this area. There's some evidence, some data that suggests that there's some hormones, reproductively related hormones that are involved with this that could signal that the environmental conditions are more favorable. We think about what we call the periparturient rise, and shortly after lambing, we generally see an increase in fecal egg count and parasite burden in those adult females. And that's attributed with, a potential decrease in immunological fitness, that increase in stress associated with that lambing time, as well as the study that I have cited here looked at progesterone levels, that reproductive hormone, and its association with this hypobiosis. And so when we think about, sheep are generally pregnant during the winter months, typically lamb in the springtime. If the parasite can somehow sense that progesterone levels are high, that could be a cue that the environment's probably not favorable for continued development. When progesterone levels decrease after lambing, then that would likely mean it's springtime, likely mean the environment's more favorable, and they can come out of that arrested development, that hypobiosis, and resume their life cycle. Like I said, there's a lot of work to be done in this area still, not well understood. There's some evidence out there that might be what is going on there, but wanted to mention this as well. And this is one way the parasite is able to continue its life cycle through those harsh winter months, when a parasite that typically likes hot, humid weather would generally not survive out in the environment. So when we get into actually managing the parasite, we first need to identify the problem. We need to figure out that it's truly parasites is what we're dealing with, or maybe it's something else. And there's no sense in spending a lot of our resources on fighting something that's not a problem. So we need to first identify that problem, identify what parasite is causing that problem, and then we can accurately determine how we're going to address that. So a quote that I like, you can't select or improve something that you don't measure. And so we need to measure traits associated with parasitism. We need to make data-driven decisions. So we need to think about what traits are associated with parasitism, what traits can we measure to help us determine if we have a parasite problem, what type of parasite problem we have, and provide us with some clues as to what we can do to manage that. And so a really simple place to start is deworming records. And I think all of us, we potentially already do this. It's something that's fairly easy to record and a great place to get started. If you're not keeping any records at this point, this is a great place to start. The pros with this, it's easy, simple records to keep. Every time you drench an animal, record what you drenched it with, who you drenched, when you drenched it, and just keep those records in a notebook, and you'll have that to go back and refer to. The cons to these deworming records, we don't know the actual parasite burden. Something is telling us that we need to deworm them, so we are, but we don't really know if the animal is parasitized or how parasitized they are. And so we need more accurate tools to help us determine that. We'll talk about that in just a second. So some things we can keep records of: what animals are dewormed, are the same animals needing multiple treatments. That could be a clue that that animal may not be as resistant or resilient as what we would hope. That's an animal that if they require multiple treatments, maybe they need to find a new home. When do you deworm? Are you deworming earlier in the season, later in the season? Are there some trends in the time of year that you deworm? What dewormers are you using? And how effective are those dewormers? And so that really brings us to these two questions. How are you determining who to deworm? Are you deworming everybody? Hopefully not. But how are you determining which ones to deworm and which ones not to deworm? And how do you know your dewormers are effective? Because there's no sense in using a treatment that is ineffective. And so we need to be able to determine if those treatments truly are effective and if we should continue using them. So first I want to talk about FAMACHA scoring, and many of you probably are familiar with this. FAMACHA scoring is simply assessing the anemia status of that animal based on the mucous membrane color around the eye. And so you can see in the photo here how the the technique is being demonstrated, and we're comparing the color of that mucous membrane to the colors on the card, and then determining what that FMACHA score is. The more parasitized that animal will be. Generally, the paler and the coloration will be, resulting from that increased level of anemia. And we can use this as an indicator trait to help us determine how parasitized that animal might be. The positives, again, it's an easy on-farm assessment. Any one of us can learn how to do it. We can carry this FMACHA card with us, and we can determine what these FMACHA scores are, and it can be a great metric to help us determine who needs to be dewormed and who doesn't. The negatives, it is only relevant for Homunculus contortus infections. This is the only worm that's consuming blood that's causing that anemia. And so really, if we're using FMACHA scoring, it needs to be for assessing, a Homunculus contortus type infection. If it's another worm that's causing a problem, we probably need to look at other things. And it is somewhat subjective. And I say that a little bit hesitantly. There's a training that you must go through to be certified in FMACHA scoring. The purpose of the training is to try to limit the subjectiveness, to make sure that people are adequately trained, that we have the skills and the knowledge to accurately determine what that FMACHA score is. So make sure you go through that training. There's numerous online trainings available for you to do, and then you can receive that FMACHA card. But we want to make sure you go through that training before you use that card. So you're going to compare the color of that eye, the mucous membrane around the eye, to the color on that card, determine what that FMACHA score is, and that's going to assist us with what we call targeted selective treatments, which we'll come back to and talk about in just a little bit. Along with FMACHA scoring, we can also use the 5-point check. And the 5-point check just looks at more of a holistic approach and looks at other areas, indicators that there might be a parasite problem, and other parasites besides just Homunculus. So not only do we look at the eye and the paling in the eye associated with that FMACHA score, but we also look at the back, we look at the body condition score, we look at the rear end of the animal for that DAG score, fecal soiling. That's a common indicator of maybe some coccidia issues. We look at the jaw, and evidence of bottle jaw, and from that hypoproteinemia. The nose, nasal discharge, nasal bots, not as common, but another area that we can look. So I have a couple pictures here, examples of what bottle jaw can look like. And again, we can use this 5-point check as a tool to help us determine who needs to be treated and apply it to this targeted selective treatments. So that's how we can determine who needs to be dewormed. But how do we know if our dewormers are effective? And this is where the fecal egg counts come in. And fecal egg counts are simply a measure of the number of strongylid parasite eggs in 1 gram of fecal matter. It's very important to understand that we can't distinguish the different types of strongylid parasites by the egg stage. So we can't just look at the egg and tell if it's Telardursagia or if it's Homunculus. They're just strongylid eggs. They're just strongylid parasite eggs. That's all we can say. Now we would assume that in many of our cases, in many of our operations, it's probably majority Homunculus, but that's not always the case. So just keep that in mind. It requires what we call larval culture to determine what those different species are. And so we can actually culture those eggs to the larval stage, and based on what those larvae look like and some measurements we take on the larvae, we can determine what species of parasite is actually present there. So we take that fecal egg count and we can do what we call a fecal egg count reduction test. And so we measure the fecal egg count at the time that we deworm, the time we apply that treatment, and then we measure that fecal egg count again roughly 2 weeks later, 10 to 14 days later, and we should see a greater than 95% reduction in that fecal egg count. If we don't see it, if we see a less than a 95% reduction, that tells us that our dewormers are not as effective as they should be, and we probably need to start thinking about some alternatives. And so this can be a great tool to determine if our deworming products are actually effective. Now, measuring adult worm burden would be great. It would be great to say, I applied that treatment, I don't have any adult worms, or this animal doesn't have any adult worms despite being exposed to infective larvae. It'd be great to be able to do that. Unfortunately, it's not very practical. The only way to measure the adult worm burden in an animal is to harvest that animal and remove the abomasum and actually count the number of adult worms there. So it's kind of like carcass traits in that way. It's just not practical. We can't collect it on the live animal. And so we have to use alternatives. And so for carcass traits, we've developed, carcass ultrasound as an effective tool to do that. With our parasites, we use fecal egg counts to be able to get a sense of what the parasite burden might be inside of that animal. These fecal egg counts are a quantitative and objective measurement. So, we count the number of eggs that are there, the number of eggs that are there, the number of eggs that are there, and it is what it is, and we get that fecal egg count. And this is an indicator of true resistance rather than what we call resilience. And the resistance versus resilience debate can be become a kind of a hot topic, and one that I'm not going to dive into too much in detail now. We can come back to it later if we have some time, but we use fecal egg counts to measure true resistance. It can be a valuable selection tool, and I'll also talk about that here in a little bit, and it is related to the number of adult worms in the gut. If you don't know how to do fecal egg counts and you're interested in fecal egg counts, there's a link here on this slide. The American Consortium for Small Ruminant Parasite Control does have some information on their site and some labs around the country that you can send these fecal samples to, and they can run those fecal samples for you, determine that fecal egg count at relatively low cost. So why don't we just use a dewormer? Why don't we just treat everything? And I hope this illustration shows that. So within a population of worms, we have male and female worms. We also have some worms in that population that are resistant to our deworming products. We also have some worms in our population that are susceptible to our deworming products. Every time we treat with a dewormer, we wipe out the susceptible population. Any worm susceptible to the dewormer gets killed. So what does that leave us with? Well, all that's left is the resistant population. And I like to say, what doesn't kill you makes you stronger. And the song always pops in my head when I think about this. But it's an important concept to remember, in that every time we use a dewormer, we need to think about that as applying selection pressure towards the resistant worm population. So when we treat with the dewormer, we're selecting for, we're putting pressure on, we're generating more resistant parasites. The more and more we do that, the greater population of resistant worms that we will have, and the less effective our dewormers will become. There are only 3 chemical classes of dewormers, and resistance is documented to all 3 classes. So we really can no longer count on chemical treatments. Now, if the situation gets bad enough, we certainly want to be able to use these chemical treatments as kind of a last resort to be able to help that animal that is sick, that has that high parasite burden. So we need to do anything we can to maintain the effectiveness of that deworming product. And so we have the 3 classes here and some popular products that fall within each one of those classes of dewormer. We haven't had a new class of dewormer in 30, 40 years or more, and so we need to be cognizant of that and know that we don't necessarily have the next great thing to use. We can't count on chemical treatment. We need to look for alternatives. We also need to know who is most susceptible. And this graph shows fecal egg count relative to lamb age. And we can see as those lambs get older, initially that fecal egg count really doesn't start to show up until the lamb is a month old or so, and this would be in a grazing type production system. Remember, there's that 3-week lag time between infection and when we get a fecal egg count. So that parasite, the fecal egg counts start to rise. It peaks somewhere around 120, 130 days of age. And at that point, the animal's got a decision to make. Either their immune system, their body recognizes that parasite infection, responds, and that parasite infection, that parasite burden decreases, fecal egg count decreases, and they do that on their own, or we treat the animal. Or the animal doesn't respond to that infection, doesn't recognize their infection, fecal egg count continues to rise until that animal dies. And so in that period, month or 2 following weaning is a period of high susceptibility, when fecal egg counts are generally going to be relatively high. Those animals, are susceptible to being infected or likely infected, and we need to be cognizant of that and make sure that during that time period, we have those strategies in place to help mitigate that infection level, or just have animals that are able to respond to that infection and do it on their own. We also need to remember that twins and triplets generally are more susceptible than singles. So we need to pay a little bit closer attention to those animals being raised as multiples as compared to our single reared lambs. So what about corrective actions? What can we do to help mitigate this and manage this? Unfortunately, there's no single tool or method that's a golden ticket. I can't tell you, do this and all your parasite problems will be solved. Unfortunately, that just doesn't exist. I wish that what did exist, but it doesn't. Rather, we need to use a combination of different tools, different management strategies to help us manage our parasites. And this brings us to that integrated parasite management, where we're looking at combining different tools in the toolbox to come at the parasites from multiple angles to help us manage this infection. So we can think about this from an environmental standpoint as well as an animal standpoint. So from the environmental side of things, we can think about grazing management. And I'll go into each one of these in a little bit more detail, so I'm not going to spend too much time here. Or from the animal side and what we can do in terms of managing those animals or treatments that we can apply to those animals to help manage the infection level. So we need to select several of these tools and put them in our parasite management toolbox to be able to effectively manage the parasite populations on our operation. So starting off with grazing management, this comes back to that parasite life cycle, and I included the parasite life cycle here in the lower right for your reference. When we talk about grazing management and how we're going to manage our forages, our grasses, our animals within that system, it's crucial that we understand the life cycle. So we're going to use temporary fencing or a variety of different paddocks that we might have available to us to minimize the exposure of that animal to the infective L3 larvae. So if we think about the life cycle of the parasite, we need to rotate those animals at least every 3 to 5 days. In hot, humid weather, maybe even need to be more frequent than that. But I'd say at an absolute maximum, 3 to 5 days. We think it takes a week to 2 weeks from the eggs being shed until we have infective larvae. So if we're moving those animals every 3 to 5 days, we're moving those animals faster than the infective larvae can develop. So by the time the infective larvae, establish on the pasture or present on the pasture, those animals have already moved to the next paddock and are grazing, fresh forage in the next paddock over and are not directly exposed to the L3 larvae that have just developed. We also need to avoid overgrazing. About 90% of our infective larvae can be found in the first 4 inches of the forage. From the ground up 4 inches, that's where 90% of our infective larvae reside. And so if we can manage our grazing system so those animals aren't grazing closer than 4 inches, we're preventing them from being exposed to 90% of the infective larvae out on that pasture. So if in doubt, move them. More frequent moving, the better. Not only is this good for parasite mitigation and avoiding exposure to the infective larvae, it's also good from a forage health standpoint. We think about a lot of our cool season forages like fescue that we have a lot of down here. The minimum grazing height, stop grazing height on fescue is about 4 inches. Fescue to keep that forage healthy and growing and well established, we don't want to graze that closer than 4 inches anyway. And so not only is it good for the grass, but it's also good for parasite management. We need to adjust stocking density accordingly. I can't give you a specific number on this. It's going to vary farm to farm based on the animals you have and the forages that you have and where you're located, but adjust accordingly based on the amount of time you want to spend in a given paddock, how fast the forage is growing, what the forage height is at the given time. You can adjust these things to make sure you're not overgrazing. And then provide adequate rest period. And this is another one, I don't have a specific number to give you. Generally, it's recommended that at least 45 days or more of rest period before we come back and graze that paddock. And I realize that for a lot of operations, for small acreage operations, this is very difficult to be able to give it full 45 days before we come back and graze. But generally, the longer, the better. We need to keep in mind, though, that the longer we wait, while it's from a parasite standpoint, it's probably good. From a forage standpoint, those forages are becoming more and more mature and the quality of those forages are decreasing. And so we need to balance providing our animals with the highest quality forage with managing parasites. And so it's kind of a balancing act there. We don't want to just feed them a bunch of mature forage that's all seeded out, but at the same time, we want to make sure that we're cognizant of the parasite burden that might be on that pasture. So keep those things in mind. We can also, after grazing, we can let that pasture regrow and then harvest that for hay. In the process of making hay and the drying process of making hay, those parasites also die during that drying out process. And so we could come through and take a cutting of hay off before we come back and graze, and that's going to remove some of the parasite burden as well. So you could also implement that as a management strategy. We can also think about the types of forages that we're selecting, and these forages that contain higher levels of tannins, like bird's foot trefoil, Serratia lespedeza. The type of forage you choose is going to depend on where you're located geographically. Here in the southeast, lespedeza is probably a bit more common. Further north, you might be able to have more success with bird's foot trefoil. But both of these, and I have graphs here from a couple studies showing the effectiveness of both of these forages in helping to mitigate parasite burden and decrease fecal egg count in animals that are grazing these tannin-containing forages. These tannins improve protein availability for the animal by binding protein at high pH, and then they release that protein at low pH. So in short, they bind up that protein in the rumen, they carry that protein to the abomasum, to the more acidic stomach like we have. When they get to the abomasum, they release that protein where it can be digested and absorbed in the abomasum and then small intestine. And so it increases the availability of that protein. It minimizes microbial digestion and breakdown of the protein. And so we've seen, anything that improves rumen bypass protein generally helps those animals to combat that parasite infection. And so feeding these tannin-containing forages is one way to do that. Another way is to just supplement more. We can just feed more. And by feeding more, we can decrease parasite burden and improve the performance of our animals. So in this study, they were supplementing with a corn soybean meal and then fish meal supplement. So they were adding that fish meal for that rumen bypass protein. And so by just increasing that supplementation amount from 1% of their body weight to 2% of the body weight, you can see the fecal egg count dropped, almost was cut in half or more. And then if we look at growth rate on the chart on the right, those animals at 1% body weight supplementation were actually losing weight compared to the animals that, at 2% of their body weight were gaining weight. And so a substantial difference in average daily gain by just increasing supplementation from 1% to 2% of body weights. And so we think about managing parasites, sometimes we just have to feed a little bit more. By feeding more, we provide more nutrients. We help that animal replenish their nutrient supply. That they're losing blood every day from the parasite. We need to help that animal respond to that and replenish those losses, and be able to effectively respond to the parasite infection. So keep that in mind, and supplementation can be a good strategy as well. Mixed species grazing. I wanted to mention this. Won't spend a whole lot of time on it. In theory, the opportunity exists here for multi-species grazing to help manage our parasites, but the data is limited. There's not a lot out there showing the effectiveness of this. But when we think about our parasites, many of our parasites are species specific. And so our sheep and goat parasites generally don't infect cattle. A lot of our cattle parasites don't infect sheep or goats. Sheep, goats, llamas, alpacas, deer do all share worms, so we need to remember that. But if we can manage a co-grazing system or rotating species in pastures, that can help us limit, in theory, pasture larval burden. You think about one species kind of being vacuum cleaners for the other. They graze, they consume larvae that would infect the other species. In consuming that larvae, the larvae dies passing through that digestive tract. So the sheep are cleaning up the cattle parasites, the cattle are cleaning up the sheep parasites. In theory, it should work, but like I said, limited data out there showing this, but did want to mention it briefly. And then we come back to targeted selective treatments. And this is where we're going to implement that FAMACHA score, and we're going to implement 5-point check to help us determine which animals need treatment. And we're not going to treat everybody. We're only going to treat those animals that truly need it. And by doing that, we're going to maintain what we call refugia. We're going to maintain a refugia population of parasites that are susceptible to our deworming product so that we can maintain the effectiveness of our dewormers into the future. And so by using FAMACHA scoring, we're going to only treat those animals that are FAMACHA score greater than or equal to 4, or FAMACHA score 3 with a poor body condition. If they're FAMACHA score 3 and they're really thin, they probably need some help and assistance. If they're FAMACHA score 3, they're fat and happy, they're probably okay. We probably don't need to treat those animals. So we can use this FAMACHA scoring to help us determine who needs treated and who doesn't. So this refugia population, I found this example was put together by the Center for Vet Medicine, and I think it does a really good job illustrating this refugia concept. And so if we look at the chart here on the right, let's look at the column on the left there. So let me get my laser pointer here. There we go. So let's look over here first. So if we look at these blue dots represent susceptible parasites to our dewormer. The red dots represent resistant parasites to our dewormer. So if I treat the entire population with a dewormer, I wipe out every single susceptible parasite. I wipe out every single blue dot, and all I'm left with are the red dots. All I'm left with are resistant parasites to breed and make more resistant parasites. If I only treat a subset of that population, so I only treat, in this case, 50% of the herd, some of those blue dots remain. Some of them get wiped out, but some of them remain. And by leaving some of those blue dots in the population, those susceptible parasites are around to breed and to propagate more susceptible genetics that then when I do have that animal that does get really sick and does have a parasite infection and I do need to treat, that dewormer is still effective because I still have susceptible worms within the population. And so this is where targeted selective treatment can be very valuable. Only treat those animals that need it. That's going to maintain that refugia, that susceptible population of worms that can propagate susceptible genetics and help us to maintain the effectiveness of our dewormer products. We can also remember the 70-30 rule and this idea that 70% of the parasites within a population are carried by generally just 30% of the sheep or goats within that flock or herd. I have the distribution here for our Katahdin lambs here on campus, and you can see that the vast majority of our lambs have relatively low fecal light counts. We just have a few individuals that have very high fecal light count, very high worm burdens. And so if we can accurately identify the 30% that carry 70% of our worms, that's the problem 30%. That's the 30% that we can think about applying some selection pressure against, getting rid of those animals, culling those animals, or just treating that 30% rather than treating the whole population. And we can be much more effective in that way. And then when we do decide to drench, we need to do that in a proper way. So we always need to provide the full dosage using the appropriate drench gun. We certainly don't want to underdose, but we also need to be very careful about overdosing because some of our dewormers are toxic if overdosed. And so it's important that we read the label. Goats generally require higher dosages than sheep. So if any of you are goat producers, if you have goats, I did want to mention that goats do generally metabolize the dewormer faster, and so we need to provide higher dosages to goats. And we need to use a scale to accurately determine weight. We don't want to just, we might be good at eyeballing it, but a scale is always best to determine what that actual weight is so that we can make sure we apply the proper dosage. And I did want to point out on the American Consortium for Small Ruminant Parasite Control website, they have deworming charts for camelids, goats, and sheep. These deworming charts are very useful tools. I use them all the time, recommend them all the time. And it provides for each type of dewormer that's out there, for every weight class of animal, what the dosage is for a given deworming product. These were developed by veterinarians, and I recommend you checking those out if you're interested on dosages. The links provided here, and they're a great resource that the consortium's put together for us to utilize. And now, as a consortium, we are generally recommending that we use combination dewormer use. So instead of just using one dewormer from just one class, we're going to choose a dewormer from each class and use those in combination. So we're going to drench with each dewormer at labeled dosage and sequence. We do not want to mix these dewormers together. Rather, we're going to draw up one dewormer, give that dewormer, drop another dewormer, give that dewormer, drop another dewormer, give that one. We want to give 2 or 3 dewormers from different classes to the animal at that treatment time. We do need to remember that not all of these dewormers are labeled for sheep. And so when we get in that extra label use conversation, we need to include a veterinarian in that. I can't tell you to do that. That needs to come from a veterinarian. And so you need to keep that veterinary client-patient relationship in mind. And again, we need to use this in a targeted selective treatment manner. We don't just want to treat everybody. We only want to treat those animals that need it. But when an animal does need it, this combination use can be a much more effective treatment method. We might have 2 dewormers that individually might only be 80% effective, but we put them together and maybe we get 95 or 90% efficacy. And so think about using these dewormers in combination rather than just using one dewormer or another. Another tool that's come out is copper oxide wire particles. And I will say copper oxide wire particles are safe to be given to sheep. It's a relatively low dosage. These can be packaged in gelatin capsules. The dosages are provided here for lambs and adult use. And these copper oxide wire particles have a negative impact on adult homunculus survival. And so they're going to take care of those adult worms and mitigate that level of parasite burden within the host. Out of fear that these copper oxide wire particles the worms might develop resistance to them, it is only recommended that we use those on Fibonacci score greater than 3 individuals. So just like we would only deworm the high Fibonacci score individuals, we only want to use the copper oxide wire particles on those high Fibonacci score individuals as well. There's not that I'm aware of, there's no documented resistance to copper oxide wire particles that I'm aware of, but out of fear that that could develop at some point, we want to make sure this remains as an effective treatment as possible. And so we want to only treat those animals that truly need it. And on that WormX website, there is another fact sheet on copper oxide wire particles that is another great resource for you. I don't have time to dive into each one of these things in full detail, and so I wanted to provide a link to more information about these management strategies if you want to look into them in a little bit more detail. And then genetic selection. And this is an area that I think has a bright future and an area that has a lot of opportunity. And so we think about genetic variation and genetic resistance to parasitism. We see that resistance, we see that variation not only between breeds, and we have some breeds that we classify as more parasite resistant and other breeds we classify as more parasite susceptible, but we also see a lot of variation within a single breed. And oftentimes, for actually many of our traits, we have more variation for a given trait within a single breed than we do between breeds. And so not only is it important to select the right breed, but it's important to select the right individual within that breed. We want to identify the genetically superior individuals within a given breed. And so I put, just to help visualize this, I have this kind of distribution here. We have our more resistant individuals versus our more susceptible breeds. Our standard parasite resistant sheep include breeds of Caribbean origin like the St. Croix, the Barbados blackbelly. Those breeds, based on their region of origin, if they weren't parasite resistant, they probably weren't going to survive. And so they've kept that level of resistance. These animals respond very well to parasite infection and mitigate that infection level. On the other end of the spectrum, we have breeds, generally our black-faced breeds are generally more parasite susceptible, although there is variation within those populations as well. And then in the middle, we have a lot of breeds, the Catawba and Texel, Dorset, just to name a few. And most of our breeds fall somewhere in between, where within a given breed, we may have individuals that are fairly resistant, and we might have other individuals that are fairly susceptible. So the key thing there is to identify which ones are the resistant ones and which ones are the susceptible ones, and then make accurate selection decisions based on that. And so what selection tools do we have available to us? In terms of phenotypic selection, we can go back to that Fibonacci scoring and those Fibonacci records. We can go to our deworming records. We can identify those repeat offenders. Who needs to be dewormed multiple times in a given grazing season? And those are problematic animals. Those are animals that probably don't need to stick around, and we can apply some selection pressure to and cull those individuals and probably make our lives a little bit better and remove some of that parasite burden from our operation. But then we can also do fecal egg counts. And fecal egg counts are really our most valuable selection tool because they allow us to identify those individuals that actually have the worms. Fibonacci scores deworming, we're just identifying those animals that are getting more anemic and needing treatment. We don't know how many parasites they actually have or how many eggs they're actually shedding on our pasture. And fecal egg counts are going to allow us to do that. And so highly recommend doing some fecals and being able to determine who those individuals are that are truly carrying most of the parasite burden. There's a chart here that I included from a fact sheet that Jim Morgan put together. It's also available on the Parasite Consortium website, but it provides a great resource there for some different traits you can utilize for on-farm selection for parasite resistance. So we can implement those good culling practices. We can identify, that 30% of individuals that carry 70% of our worms. We can apply some selection pressure to those individuals, get rid of those individuals, and mitigate some of our parasite problems in that way. It's important to understand that those sheep need to be challenged before we can get an accurate fecal egg count. Remember, it takes 3 weeks from the time the infective larvae are consumed until we can get a fecal egg count. And so there's that 3-week delay. So we need to time our fecal egg count collection for generally 4 to 5 weeks after known parasite exposure. The goal is to have an average fecal egg count in that contemporary group in the animals that we sample of at least 500 eggs per gram. And that's going to tell us that there is adequate level of exposure, an adequate level of infection to determine, who's the more resistant individuals versus who are the more susceptible individuals. So just a quick review of the life cycle and the timeline here. We have those infective larvae. In that first week, they're going to develop into the L4. Remember that the L4 feeds. It's not sexually mature, but it does feed. And so we can actually have those sheep die of what we call acute hemoncosis within a week of turning those animals out on fresh pasture and never have a fecal egg count. We find them dead one day. They can become, if they find the wrong patch of grass and consume a lot of infective larvae, those infective larvae all mature to L4s at the same time, all start consuming blood at the same time. They're not producing eggs, but they're consuming blood and causing anemia, and then that results in potentially some dead individuals. And so we need to be aware of that. It's going to take 2 to 3 weeks, on average about 21 days, before we get mature adults that are going to begin shedding eggs. So then we want to time our sample collection for 4 to 5 weeks after known parasite infection, or exposure, I should say. And that's going to make sure those animals have the opportunity to become infected if they are truly susceptible, and then either mitigate that infection, which are the more resistant individuals, or allow that infection to establish, which are the more susceptible individuals. If all of our individuals, if they all have fecal egg counts of 0, that doesn't really tell us much. It could mean that they're all resistant, but it could also mean they just weren't exposed to any parasites. And so we need to at least make sure that they have the opportunity to be exposed, to be infected. They need to be challenged to get an accurate fecal egg count. So we have those records, we have the phenotypic selection, but then we also have what we call estimated breeding values, which I think many of you probably are familiar with or have heard before. And these estimated breeding values are an opportunity for quantitative selection, and they allow us to measure individual genetic merits. So the actual genetic merit of the individuals rather than just the phenotype. And these estimated breeding values combine individual performance with pedigree information. How well did mom and dad do? What are the genetics of mom and dad? Any progeny records that might be available once that animal starts producing progeny. And then new for Katahdins, as of this past fall, we now have genomic information that's included in that genetic evaluation. So we have what we call genomic enhanced estimated breeding values, which improve the accuracy of those breeding values. And because there's so much information that's included in these breeding values, they are the best tool in our toolbox in terms of selection tools that we have available. So like I said, they're predictors of genetic merit. They're a numerical representation of genotype. So it's simply a number that tells us what the genotype is of an individual. And it's going to simplify selection practices in that way, because I can look at a single number or a couple numbers and be able to understand the genetics of that individual without, overly complicating it. And because there's so much information that's included in these breeding values, because it includes individual data, pedigree information, progeny data, genomic data, these breeding values are more accurate than our raw performance data. They're more accurate than adjusted performance records, and they're more accurate than RAM tests. And so really these are the best selection tool that we have available to us. So remember that phenotype, what we observe in that animal is a combination of the animal's genetics plus the environment that they're exposed to. That environment is not passed from farm to farm, from region to region, from generation to generation. Environment doesn't stay constant. The genetics, though, if I buy a ram from another state, the DNA is the same. I'm bringing that same DNA here. So that genotype can be passed from farm to farm, from generation to generation. And so the key is accurately determining what that genotype is, and that's what breeding values do. So those breeding values, you can really think about it like this, where phenotype is a result of the breeding value of the individual plus the environment that they're exposed to. So they remove that environmental component, and they allow us to just assess what's genetic. Fortunately, we have a breeding value for fecal egg count. So instead of just looking at those phenotypic measurements of fecal egg count, we can actually look at the breeding values and the genetics associated with fecal egg count. This tool was developed at Virginia Tech in the early 2000s by Dr. Notter's lab. The heritability is moderate, 20 to 25%, so similar to a lot of our growth traits. And it's reported as a percent change, so negative values are good. We want to reduce, we want to lower fecal egg count. And so, lower fecal egg count would indicate a more parasite-resistant individual. So within any population, we have the majority of individuals exist around breed average. We have some individuals with extremely low fecal egg count breeding values, other individuals with extremely high fecal egg count breeding values. And if we can accurately identify and select for those low fecal egg count breeding value animals, we're going to improve the parasite resistance within our population. Quick example of that, we have two rams here. Ram on the top has a negative 60% fecal egg count breeding value. Ram on the bottom has a negative 10% fecal egg count breeding value. An individual only passes half of their genetic merit to their offspring. And so we go from an EBV, an estimated breeding value, to an expected progeny difference. We simply divide by 2. And so the ram on the top, the progeny difference would be negative 30%. The ram on the bottom, the progeny difference would be negative 5%. The difference between those two is 25%. So we would expect if these two rams were mated, hypothetically the same group of ewes, we would expect progeny from the ram on the top to have a fecal egg count 25% lower than progeny sired by the ram on the bottom. And so with this, we can slowly, over generation after generation, we can lower fecal egg count and improve the parasite resistance within our populations. Just showing the distribution within the catodons and the range there. There's some catodons that are, negative 100% on fecal egg count EBV. That's 100% reduction. That's the most you can reduce something. So those are the most parasite resistant individuals within the breed. We also have individuals within the same breed of sheep that are, up over 200 to 300% on fecal egg count. So that's, a range within just the catodon breed alone. We have a range of, over 400% on post-weaning fecal egg count. The breed average right now is sitting at about negative 30%. And if we look at the catodon breed over the last 10 years or so, the catodon breeders have identified this as a trait of importance, selected for it, and they have lowered fecal egg count. The breeding value, the genetics for parasite resistance, they've improved those parasite resistant genetics over the last 10 years by lowering that fecal egg count. And so there is evidence here that it works and it has been effective for these breeders, and they continue to select for it. So I think genetics can be a very valuable tool in the toolbox. If we can just make our breeder animals to be more resistant, I think that's really ideal. And then we can add things in on top of that. But I think genetic selection can be a big opportunity. I did want to mention biowerma. That's been another hot topic. Biowerma, it's a fungus, Dunningtonia, that disrupts the parasite life cycle at the larval stage. So it actually, we feed it to the animal, but it actually passes through the animal's digestive system, doesn't do anything to the parasite inside the sheep. So passes through the parasite, passes through the sheep, in the feces when those L1 larvae hatch. This fungus attacks those L1 larvae and kills those L1 larvae, resulting in lower pasture burden, lower pasture contamination, and consequently fewer opportunities for reinfection. So keep in mind, this is not a treatment for parasites, but it can be used as a mitigation tool by limiting the amount of larval burden that may be on our pastures. And then the opportunity for reinfection is decreased. Did want to mention, the dosages here based on body weight and the associated cost per head per day. Keep that in mind if you choose to use this product. I just wanted to mention this is it's been a common topic of questions in recent times. And then lastly, confinement housing. If we just can't get parasites under control, if nothing is working, we can move those lambs to a confinement housing type situation. These gastrointestinal nematodes generally cannot complete their life cycle in a dry lot environment. Unless they're eating a lot of their bedding, the opportunity for reinfection is really not there. And so we can get the parasite burden under control by just getting them in a dry lot, getting them inside, and getting them away from the worms. We need to consider the cost of the buildings, feed, and labor associated with this. But at the same time, we need to think about opportunities on the revenue side of things in terms of improved animal performance, improved weight gain, and marketing quality of those individuals that are raised in this type of housing system versus out on grass. So some things to think about, not telling everyone that this is what they have to do, but it could be not, if things get bad and there's nothing else you can do, it sometimes is an effective tool to just, get them away from the worms altogether. So we have a lot of tools in our toolbox, a lot of tools that are available to put in our toolbox. We need to create a plan. We need to think about what is practical, what can be implemented on your farm. Again, we can come up with the best parasite management plan, but if it can't be practically implemented, then it doesn't do us much good. And so think about what tools can actually realistically be implemented. Use a combination of different methods. This is not just a one tool, fits all. It's not, there's no golden ticket. So you can't just pick one of these and think it's going to work single-handedly. We need to think about using a combination. So multiple different tools and how those tools are going to work together. So just as an example, here at NC State, our small ruminant unit, our parasite management plan includes selection of rams based on the fecal egg count EBV. We measure fecal egg counts multiple times in all of our lambs and identify those more parasite resistant individuals. We rotate our pastures frequently and minimize overgrazing. We're not moving pastures every day, but I'd say on average, we probably move a couple times a week. We only treat those animals when absolutely necessary, and we do provide some nutritional supplementation. We know those animals are going to experience a time of higher stress, that are going to be more susceptible. We do provide some supplementation. We do provide, in certain cases, some confinement housing to help mitigate those problems so that we can make sure those animals stay healthy and continue to perform. And then lastly, we need to evaluate the results. We need to think about, are our tools effective? We need to continue to monitor parasites. We can't just implement these tools and then forget about everything. We need to continue doing FAMACHA scoring. We need to continue with the 5-point check. We need to continue with the fecal egg count and the deworming records. Even after we implement these things, we need to continue to monitor for parasites to show, to make sure that those tools remain effective. So we need to use those data to make decisions. Are the metrics improving? Are we lowering fecal egg counts? Are we decreasing our deworming incidents? If yes, keep to the plan. Those things are working. Let's keep, keep doing those things that are working. If it's not, if we continue to have deworming problems, we continue to have high fecal egg counts, we need to look at other options. Maybe we need to add some tools to our toolbox. Maybe we need to do something differently to address those challenges. But I really want to emphasize that after we implement these tools, we need to continue to monitor, to continue to evaluate these results to make sure that the plans that we put in place do remain effective and that we are managing the parasites with those tools. So with that, I apologize that we ran a little bit over here, got long-winded, but I'll open it up for any questions now. And
yeah, thank you, Andrew. There's a lot of good information, and we've had a lot of questions come in while you were talking, and I'll get to as many of those as I can here the next 20 minutes or so. Just a reminder, the question box is there that you can type questions into. If you feel the need or to actually talk to Dr. Weaver directly, you can raise your hand, and if you do that, I will see that and call on you by name and unmute you, and you can go ahead and talk to him directly. But let's get to the ones that were typed in during your presentation first. And I'm going to take these kind of in the order of your slides there. So although not all of them really have to have a slide in front, they're pretty general. But we had somebody asked, they said that they have taken fecal samples to several different veterinarians and they're always told strong giles. How important is it that we get a more specific diagnosis, and how do you suggest I go about doing that?
So like I said, you can't differentiate, you can't determine the species of the parasite based on the egg stage. So you're stuck with just the strongylid parasites, the strongyles. You know that they're in the family. So that could include homuncus, it can include Teledrossagia, the Trichostrongylus species. And so to go into more depth to determine what species you actually have, you're going to need to work with a lab, and your veterinarian may be able to do it. They may not be comfortable. If you're interested in determining what those species are, I suggest reaching out to one of us in the American Consortium for Small Ruminant Parasite Control, and we can try to get you connected with a lab that can help you with that. But like you're not, the vet's not going to be able to necessarily, without culturing those eggs, culturing those larvae, they're not going to be able to tell, what's homunculus versus what's Teler based on the egg stage. They're just going to be able to tell you that they're strongylid eggs that they observed. And so there does need to be a little more work to be done to determine what those different species are. In general, there's going to be a lot of homunculus there. There could be some other ones there as well. So I don't know if that exactly answered your question. Unfortunately, I don't have a perfect answer there. I would suggest reaching out to one of us and we can try to get you some more information.
Okay, and in regards to the FAMACHA scoring, somebody made a comment, really, the biggest negative to FAMACHA scoring for me is that it needs to be done as often as every 2 weeks, which is impractical in a pasture management system. I don't know if you have a comment to make on how often that needs to be done and maybe some practical tips on implementing it well.
Yeah, I mean, I would agree, especially during kind of the height of the grazing season, the height of the parasite season. I would agree with you that, at least every 2 weeks is when I like to check on, check FAMACHA scores, check on how the sheep are doing. Because I've seen sheep that, 1 week they look pretty good, and 2 weeks later they're in fairly rough shape. Like I said, the L4s feed, and we don't necessarily have a fecal egg count, and so the L4s can, develop, within a few days of consuming infective larvae, and so we can go from a fairly healthy individual to a fairly anemic individual in a relatively short time period. So we do need to check FAMACHA scores frequently. Unfortunately, I would agree with you on that. And I realize that it's not always practical to do that. And so if you aren't able to get those animals in on a regular basis, then that, further emphasizes the need for some of these other tools. If FAMACHA scoring and that can't be part of that toolbox, then think about, maybe you need to put a lot of selection pressure on parasite resistant sheep, just breeding sheep to be more resistant to the worms. If you can breed them to be more resistant to the worms, then the sheep can more likely handle it on their own without your intervention. And so, using genetic selection and finding some rams that are more parasite resistant may be a valuable tool to you to help mitigate that parasite infection without having to round those animals up every 2 weeks and FAMACHA score them. Not that, I'm not going to say FAMACHA scoring is still a very valuable tool. If you can bring them in every 2 weeks, fantastic. I'm not going to tell you, no on that. That certainly would be encouraged if you are able to do that. But if you can't, then genetic selection could be a valuable tool to you.
Great. And we had a couple of questions come in regards to copper. So the first one was, could you talk about the role of copper bolusing in helping manage barber pole? What does the latest research show, and what dosage? How long do the results last? I don't know if you've had any experience with that or not.
Yeah, so the dosages were provided on that slide about halfway through. And out of fear that I'd misspeak, I don't want to, the slides will be available and I would suggest referencing those. I don't want to misspeak here. I'm not looking at the slide directly, but the research has shown they are effective against the adult worm. So by treating with those copper boluses, you are targeting that adult worm, the adult worms that are in the gut of the animal. And so by treating with the copper oxide wire particles, you're going to remove some of that adult worm burden and minimize the burden in that way. So they can be an effective treatment method. And but remember, we only want to treat those animals that do need treated. We don't want to just treat everybody with copper oxide wire particles. We only want to treat those individuals that are FAMACHA score 3 or greater. Those individuals, just like we would with our deworming products, just those individuals that need the treatment. There's a fantastic fact sheet on the wormex.info website that goes into all the details of copper oxide wire particles. And I highly recommend looking at that. If that's a tool you want to use, read over that. That's going to provide a lot more detail than what I have time to cover this evening.
Okay, we had a couple of questions come in when you were talking about susceptibility. On the slide where you had who's most susceptible and the twins on there, somebody said, why are twins and triplets more vulnerable?
I think it comes down to just nutrient availability. Twins and triplets, they're sharing. They've got one mother that they've got to share, and we know that the weights, the growth of twins and triplet litters is going to be less than singles. You know, we're all used to that big single that, you know, has all the milk for him or herself and grows really well and has all those nutrients available, where the twins and triplets don't have that. So they're always a little bit further behind. And so I think comes down to, you know, those singles have more nutrients available to them. They're going to be bigger, they're going to be a little bit stronger, and they're able to manage that parasite burden better than the animals that are raised as multiples.
Okay. And then somebody asked a specific question on a dewormer. What about flukiver dewormer for barber pole? Do you know what I'm talking about? I'm not familiar with it. But,
yeah, I'm not. I might have to see it spelled to recognize
it's
it.
Flukiver, and I'm pretty sure this listener is in Canada. So.
Okay. So the availability of deworming products to you might be a bit different than us, you know, based on what's available in different countries. I'm not a veterinarian, and so I can't tell you specifically what deworming product to use or what dose to use it at. That's something to reach out to your veterinarian on and consult them. I can reference the deworming charts that are on the wormex.info website as a great resource, but if the dosage isn't on there, then you're going to have to reach out to your veterinarian to find out more about that.
Okay, and on the slide where you had the proper drenching, I think it was slide 26, you had some charts on there. If you wouldn't mind going back to that, somebody asked about the website that's available. So
yeah,
we could go back to that slide and show that to folks.
Right here.
There we go, right there.
wormex.info/dewormers.
Okay, thank you.
And at that same wormex.info website, you're also going to find the information about the copper oxide wire particles.
Okay, excellent. Let's see. Do you find more homunculus resistant animals tend to be more susceptible to coccidia? And if so, is it better to go for the happy medium for selection?
That's a very interesting question. There's not a lot of published data out about that. But what we've seen with some of the flocks that we've worked with and some of the little bit of work that we've done, sheep that in the populations that we've worked with, some of the sheep that are more resistant to the gastrointestinal nematodes sometimes have higher coccidia counts. And the animals that are maybe more susceptible to the gastrointestinal nematodes have sometimes lower coccidia counts. Not always the case. I can remember one particular example of that. So I'm not gonna, and that I'm aware of, there's very little published data on that. So I'm not gonna, you know, state it as that's the way it is all the time everywhere. But it could potentially exist. And so just think about, you know, what parasites, and that goes back to, you know, on that early slide early on, identify what your problems are. You know, if you're having homunculus issues, then you know, deal with homunculus and try to get that under control. If you're having coccidia issues, then, you know, focus on coccidia management. You know, sometimes it's multiple worms at the same time, and you have to do a little bit of both. In terms of published research data that we have available, we have the accurate selection tools to generate, to produce, to breed animals that are more resistant to homunculus, to breed animals that are more resistant to the gastrointestinal nematodes, the strongylid parasites. We do not have those selection tools available for coccidia at this time. And so I can't comfortably say that use this tool for selection for coccidia resistance. Just can't say it, that tool doesn't exist. We don't have that available to us, but I can say that we have the selection tools for selection for strongylid parasite resistance, and we can use that to make animals that are more resistant to the strongylid parasites.
Okay, and along those same lines, is the fecal egg count EBV positively or negatively correlated with other traits of interest? Of course, that's a loaded question, but you want to talk about any trade-offs that might be known there?
Yeah, that's a very loaded question. There's not a strong correlation between fecal egg count and other traits. And And what I mean by that, if you select for fecal egg count EBV, you're not going to drastically change anything else. You're not going to go way off into some direction you don't want to go on another trait. There are some weak, moderate relationships between the two, but they're all small enough to where with observation, with accurate selection decisions, you can make progress in both in the direction you want to go at the same time. So hopefully, I hope that answered the question.
Yep, I believe that does. So someone asked if you could clarify a little bit on what does the lower fecal egg count actually mean? And what they mean is that although the same number of L3 may have been ingested, those L3 did not mature and produce eggs. You want to talk a little bit about that?
Yeah, so I could do a whole nother hour-long seminar on that. Yeah, so that's a really loaded question. In short, those animals that have lower fecal egg counts are animals that, in theory, their immune system was able to recognize and respond to that parasite infection, minimize or prevent parasite establishment within the gut. And as a result, if the parasite can't establish in the gut, they can't make their home there, then they're not going to reach maturity. They're not going to produce eggs. We have a lower adult worm burden. We have a lower fecal egg count. And in our most parasite-resistant breeds, like the St. Croix, their immune system is so effective against the parasite that they recognize that parasite within the first, couple days of infection. They respond to that parasite very quickly, and they minimize establishment of the parasite. And so we have very, very low adult worm burden and consequently very low fecal egg count, despite the animal being exposed to those L3 larvae. Where our more susceptible breeds or more susceptible sheep, those animals, they're exposed, they ingest a bunch of L3s. Their immune system has a delayed response. It doesn't recognize the infection right away. It's about a 4-day delay in response compared to our parasite-resistant breeds. So they have a delayed response, and because the response is delayed, it gives the parasite time to set up camp within the gut. And once the parasite's set up and established within there, it's made its home, it's happy. It goes on, continues development to the adult stage, begins shedding eggs. Now we have a fecal egg count. And so that's probably the shortest I can explain the differences between our parasite-resistant, parasite-susceptible individuals as it relates to fecal egg count. But that's a great question and one that we could talk for quite a while about.
Okay, excellent. And we've had 3 different mixing questions coming in. A couple of them pertaining to biowarma and mixing it with minerals, and then a general question on mixing dewormers together. And I think you mentioned that in your talk on one of the slides, but if you could expand a little bit on the idea of mixing treatments. There you go.
Yeah, I want to be very clear. Do not mix the dewormers physically together. You're going to give them at the same time. That animal standing in the chute next to you. While it's standing there, you're going to give that animal 3 drenches. Well, in this time span of a couple minutes, you're going to give them 3 drenches, but you are not going to physically mix the dewormers together. Those dewormers have chemical properties that we do not want to mix them together before giving them to the animal. And so if we're going to use a combination treatment, and we actually have on the WormX website, we have a new video series called WormX Live, and it's short video segments, recordings of some of these best management practices. And we actually, our very first WormX Live video was on this very subject. And so you can check out the WormX Live website. There's a link to that video, which can help, provide some more information on this as well. But what we're going to do, if we want to use a 3 combination treatment, I'm going to draw up a drench of the first dewormer. I'm going to give that. I'm going to draw up a drench of the second dewormer. I'm going to give that. I'm going to draw up a drench of the third dewormer, and I'm going to give that. So there's 3 separate drenches, but they're given, in a 1 or 2 minute time span or less while the animal is standing right there in the chute.
Good. And we have a question on, have you seen any research that backs up the thought that having young stock off grazing until they're 6 months of age creates an animal more resistant to parasites throughout their lifetime?
I've not seen the data. We're working on a variation of that right now. I actually have a student that's working on some of that. It's not specifically that question, but it's something similar to that. And so we might have more information for you later this year, early next year.
Okay, and another similar question, how long do nematodes live inside sheep? In other words, how long do you need to dry lot adult sheep to eliminate the intestinal worm population?
So the adults can live there for a few months, 3-4 months, sometimes maybe a little bit more than that even. So they can be there a few months, but keep in mind, if they're in a dry lot, you can go ahead and treat them because they're not going to deposit resistant eggs on the pasture for reinfection. And so if they're in a dry lot, you can treat those animals, you can remove that infection. Make sure, the key is take that fecal egg count before they go back to grass. Make sure that you've eliminated that parasite infection. Make sure, take the fecal, confirm a fecal egg count reduction, confirm that the dewormers worked before they go back to grass. But in a dry lot, if they're shedding worms that are resistant to our dewormers, there's the opportunity for reinfection is minimal, and so the chance of consuming those resistance larvae is going to be minimal. And so you can treat with a little bit more ease in a dry lot type environment than you can out on pasture. But I will stress that before they go back to grass, make sure you do that fecal egg count and confirm that fecal egg count reduction.
Okay, so so moving away from the treatment, so to speak, and more management remedies, I've got a few questions here grouped together. What are some of the more natural remedies, such as garlic? Are they effective? Want to talk about any natural remedies?
Yeah, I haven't seen anything that shows that they're effective.
Okay. Somebody asked about utilizing 4 pastures with cows, sheep, and goats. Is there a particular way to utilize pasture rotations to mitigate infection?
Yeah, so in theory it would work. Now remember, sheep and goats share worms. So same parasites infect sheep are going to infect the goats, vice versa. So just a sheep-goat rotation, not going to have any effect. Cows and small ruminants generally do not share the same parasites. So in theory, that could work. Now, if you actually go and look in the literature and find data to support that, it's very minimal. And so there's not a lot of data out there, but in theory, we know about parasite biology. In theory, it should work. But unfortunately, I don't have like the graph to go and show you that here are the numbers that prove that it works.
Okay. And along those same lines, is it helpful to use annual forage for grazing as opposed to perennial pasture?
Yeah, it can be. Sometimes those annuals, they grow a little bit taller, so they're going to be further up away from the ground. The animal's not grazing as close to the ground, and so you get that animal grazing up a little bit higher, where the chance of exposure to larvae is a bit lower. So, from that sense, they could be effective in helping to mitigate exposure.
Okay, any further research on vaccines, and are they effective against several species?
Not that I'm aware of. I know there's been some work done in the past. I'm not sure exactly where we're at right now. And so I don't count on a vaccine right now. Look for other tools. Maybe someday, but no guarantees. And I don't have any more information on that.
Okay, and I guess we got time for just a couple more questions here. What about meat goats? Are there much breed difference in parasite resistance, and how do breeds rank? In particular, dairy goats, are there breed resistance or breed differences? Sorry.
So I don't know the answer on the dairy goats. On the meat goats, most of the data that I've seen, the boar is going to be the most susceptible. There's not a whole lot of differences between Spanish, Kiko, myotonic. They're going to be a bit lower than the boar. Generally, in most of the studies that have been done, the boar generally has the highest fecal egg counts and generally shows the most susceptibility, and the other breeds are maybe a bit more resistant. Not as much work has been done on the goat side of things, particularly with inbreed. With sheep, we have data to show that there's a lot of variation within breed. I assume the variation exists within goats, but we don't have that quantified yet. And so more work needs to be done on the goat side of things, especially in terms of genetic selection and identifying those more resistant individuals within breed.
Okay, one more natural question. Any further research on dried papaya seed to reduce fecal egg counts? And this is from a producer in Florida.
Not that I'm aware of. I'm not familiar. I'm not sure.
Okay, and we'll finish up with a question on treating parasites in pregnant ewes. Any specific suggestions there?
Yeah, so just be cognizant about our dewormers and what dewormers can be used in pregnant ewes and which ones can't. Be sure to always read the label and be cognizant of that. Think about what alternatives we can use in those pregnant ewes, and a lot of things we talked about tonight can be implemented in our pregnant ewes, our late gestation ewes. And so, we can think about, maybe some supplemental feeding. Hopefully might need to be supplementing them anyway, especially during late gestation, to make sure we don't run into pregnancy toxemia issues. And so, things like that, supplementation, just general genetic selection to make those animals more resistant, good grazing practices, moving them frequently, minimizing overgrazing, things like that can be utilized certainly in those pregnant animals to minimize the exposure in those animals.
Okay. And I forgot to ask you the composting question that I've asked you earlier. And that is composting sheep waste. Does that sheep waste and waste hay, does that contain parasites because they use composted material and spread it on their pastures? Anything to worry about there?
Yeah, so if it's truly composted, and you can always take temperatures and make sure you have a good compost pile and make sure it's active and going. If it's truly composting and you're getting the elevated temperatures in there, you should, with the proper time and heat, you should kill those parasites. Now, if you're just taking the feces straight out of the barn and spreading them out on your pasture at ambient temperatures, you're certainly going to have a worm problem. But if you're truly composting it and it's heating up and it's staying at an elevated temperature for a while, you should be killing the worms by doing that. And like making hay, when you make hay, you dry out that hay. The parasites are going to dry out with the hay and generally die, and so we're not going to have infective larvae that are alive in our hays. So we can feed hay and not really have to worry about that.
Excellent. Well, with that, I think we'll go ahead and wrap up. And once again, I want to thank you, Dr. Weaver, for very informative presentation and excellent answers to a whole bunch of questions. As you can see, there's a lot of interest in this. Of course, this is a big management issue for a lot of sheep producers throughout the country. I want to thank everybody for joining us tonight and for submitting those great questions. Sorry we didn't get to all of them. We did get to most of them in some form or another, but we'll try to follow up with you with anything in particular where we thought we left some answers a little short on specific questions. You will be receiving a follow-up email to this. There is a short survey when you log off. Please fill that out. That helps us both get ideas for future webinars and get ideas on how to improve our presentations. And then also that follow-up email will go out with a link to the recording and a link to these slides, which, as you see, are full of great information. We really appreciate Dr. Weaver sharing his expertise with us tonight, and sharing the slides with the public so they can refer to them in the future. So with that, I bid everybody a good evening, and thank you again, Dr. Weaver, for taking the time to share your knowledge with us this evening.
You're welcome. Thanks for having me, and everyone enjoy your summer and good luck managing your parasites.